Hello, everyone. Thanks so much for joining us today. I am Denise Nicol. I'm the Director of Strategy for Think Regeneration. And today we are so excited to have Dr. James White from Rutgers University here with us for a Q&A with Think Regeneration's founder, Ryan Slabaugh. Ryan is the former executive director at Acres USA, which is where he first crossed paths with Dr. White. And Dr. White's research into the rhizophagy cycle is so critical in helping us producers understand how to make nutrient density possible in a biological system. So without further delay, I'm going to pass it over to Ryan to get the conversation started.
Thank you, Denise. I appreciate the chance to connect again with Dr. White. We're here for the third time together since we started Think Regeneration in 2022. And each year we made a virtual pilgrimage into his lab to kind of see what he's been working on and his discoveries. I was one of the many who first heard Dr. White around 2020 or 2021 at the conference talking about the rhizophagy cycle and that network of microbiology and fungi and biology and chemistry all working together above and below the soil, and it really helped balance out the very chemistry heavy conversations that we were having in farming for years and years and years, and it provided some missing links and missing pieces into the systems of like why these things were functioning at a higher level when we started to take care of our soil health. And so you can find our previous talks on our YouTube channel. We won't go quite all the way in the deep dive into the rhizophagy cycle like we did previously, but we'll give some updates today, and we'll try to figure out what's new, what do we need to understand. We'll also talk to our growing audience of healthcare professionals and help them really understand what they need to know about soil health and soil biology and soil microbiology as they pursue nutrition and as they pursue better access to food for their chronic disease treatment programs. So, Dr. White, thank you for being here today. And again, we're really here to really understand the mechanics of microbiology and how it behaves in the soil. Would you be able to give kind of a brief description of the rhizophagy cycle for those who might not be as familiar with it?
Yeah, no, I'd be happy to do it. In its simplest form, we have, I'll say first, because you kind of mentioned it, is that, we've believed that chemistry was the only way that plants obtain nutrients in the past, even though we knew that plants are cultivating these microbes in the roots. We still thought that chemistry is the way they get their nutrients. It's curious, and the reason for that is that no one ever saw microbes being absorbed into roots, and that's the rhizophagy process, and that is actually what plants do. I mean, they do absorb nutrients chemically. There's no doubt about that, chemical nutrients, but they also will all these exudates, sugars that they excrete from their roots, they put out of their roots, are actually attractants and nutrients that are meant to cultivate the microbes in the soil and to attract them to the root. And then when they get them in the root, or they get them on the root, right at the root tip, they will absorb them into the root cells. And this is really the thing that has been the mental barrier for people, is that if they ever saw anything, little particles in roots, they assumed, oh, those are just natural, vesicles in the roots. But actually, at the root tips, those little vesicles are microbes. They're bacteria, usually bacteria, sometimes algae, sometimes fungi, that these plants are absorbing into their cells at the root tips. And then what they do once they get them in is they'll hit them with reactive oxygen. And this is just oxygen that's highly potent, like, for example, superoxide. That's one of them, very potent form of reactive oxygen. And that will oxidize off the cell walls, off of the bacteria. And it will also, some of the bacteria will break them down completely. And so the plants are getting nutrients from these microbes, from their cell walls of the microbes, but also from some of the microbes that are fully degraded. And those nutrients are typically, they are micronutrients of various kinds, usually divalent metals that plants may have difficulty getting otherwise, so they can absorb them directly get those metals out, get those nutrients out. And then there actually is a cycle, the rhizophagy cycle, as Ryan mentioned, is where the microbes, some of the microbes, once taken in, the plant will get the nutrients out of the cell walls, and then the microbes that are inside those cells will trigger root hair elongation, and they'll be inside those root hairs, the tip, causing those root hairs to grow, causing them to elongate. And as they elongate, they're putting out the microbes from the tips of the hairs. And so once they're put out at the tips of the growing root hairs, they reform their walls, they go back out, they reform their flagella, they go back out into the soil, they swim out there, they acquire more nutrients. Later, they're attracted back to the roots. That's the rhizophagy cycle, the full cycle. But some microbes are absorbed and just fully degraded for the most part. And that is, you could just call, well, maybe that's not a cycle, that's just rhizophagy. So there, and rhizophagy, I should say rhizo, root, phagy, eating. So root eating, that's basic, simple concept where plants will absorb microbes and degrade them inside the cells. And that basically is rhizophagy. When plants do it, they show increased amounts of micronutrients in them. They show increased amounts of phenolics in the plant, phenolics that are antioxidants, and it's good for the plant, or antioxidants in the plant, or antioxidants in our bodies. They'll show more terpenoids in some cases in the plant. They'll show more. Basically, they'll express more genes. The more microbes they take in, the more gene expression the plant shows. And so you get plants with more amino acids, plants with more different kinds of amino acids, and so forth. You get a real difference. And where we see it is in, recently we've been studying this landrace corn that has a lot of microbes in it. With Walter Goldstein at the Mondawmin Institute, where he's been trying to
Wisconsin,
breed
right?
Wisconsin, yeah, at Wisconsin. And what we see is just what I said. On the lines where they're grown with microbes, you see much more gene expression. It's really, so crops become enhanced in their value, in their food value and nutrient value. More nutrient dense, basically.
I appreciate you making that link. And so, to help the healthcare professionals who might be new to this idea, when we talk about why it's important to pay attention to sourcing and where your food's coming from in your food as medicine programs, and sourcing does matter and will either catalyze or hold back the progress you hope to make if you're not asking the right questions. And last year you talked about, like, when we and some of the comparisons between gut health and soil health is that when we sterilize the systems to get rid of life, we actually prevent that it from functioning optimally overall. And so, are there other parallels that we can make between those systems?
Well, yeah. I mean, the gut microbes. If you look at the soil, the roots, the roots of plants are really filled with microbes, and also surrounded by microbes. That's the whole point. So it's about microbes. And if you look at the human gut, it's the same thing, filled with microbes. Those microbes play important roles. They break down food. They will release vitamins into the system. Well, the root and the gut are analogous systems. They have similar function. They're all about the nutrients. And really the roots are full of those microbes. Same with guts. They're full of the microbes.
Makes sense. So when we spray or we till and we do damage to those, and then we actually have to restore that biology back in the soil before we can expect it to digest again. And be optimal again. And I think when we talk to healthcare professionals, that's a big leap for them. That is a common place where we go, because that's very similar to, say, a cancer patient who's going through an extreme cancer treatment and getting their entire gastrointestinal tract, really scrubbed, and they can't just digest food anymore. And so it's very similar, where when we take entirely synthetic farm system, doesn't have the ability to actually process organics that are within it at some point. And so, um, I appreciate you making that connection as well. Uh, is there anything new that you guys have discovered in the last year that you feel like is pertinent to this?
Well, I'll talk about that, but I do want to
Yeah,
say something else about
yeah, sure.
what we were talking about. If you look at native, say, people that are hunter-gatherers, living by eating a lot of plant matter, what you see is they have very diverse gut microbiota, uh, and you compare that to modern people, who live, Western
Mm,
people. We eat sterilized foods. We grow our crops in essentially um, soils that are where the microbial community has been decimated because we've destroyed the diversity because of how we grow the crops. We turn the soil. We'll um, leave them without plants growing there, which would cultivate the plants would cultivate the microbes and increase the microbiology, but we leave it fallow for a period of time, and we treat it with fertilizer. So we essentially are negating the value of what few microbes are left in the soil. You compare the guts. We have a very uh, uh, small diversity, or little diversity, in our guts compared to these hunter-gatherers. And when we go and we consume the foods in the wild that they consume, our diversity increases and the health of the gut increases. And we know what happens when ultimately the we lose the diversity of the gut microbiology. Is we have illness, we have problems, we have gut we've got gut diseases that develops, and the IBD, for example. And the solution, of course, is increase the diversity. It's a modern invention, chemical agriculture. It's modern, and in a sense, it's still an experiment. We're experimenting with our own biology by doing that without really knowing what what it what the long-term consequences are. But we do that with a lot of things. And now, what was the question? What is new? I can
no,
tell you what's new.
okay, yeah, please do. And I think, yes, there's nothing conventional about our conventional farming systems. You're exactly right. So
No,
that's
there's
the irony of
nothing. It's an
it.
experiment. It's an experiment. We humans never did this before. We've been doing it for what, 50, 60 years, something like
Yeah,
that. But it's an experiment, and we're seeing lots of, nobody knows what causes, the ADHD and the various, but we do know there's a gut brain connection, right?
yeah.
And it's entirely possible that some of the diseases that we're seeing now in people relate to the way we're growing our crops. That's not, I can't say for sure that's the case, but it's entirely possible that that is the case. And other issues, long-term issues, all the allergies that people are getting, and it very I my I can't say likely, but I suspect it relates to the absence of microbiology in our guts, and it's that brain gut connection again that causes that. Uh, so okay, so that's the deal. But what is new for us, right? What is new for us?
Yeah.
We are fascinated. I'm fascinated by the the effect of microbes on the genetics of plants. And just to ground it a little bit, when we study microbes going into roots, or we study the microbes going into the shoots, because they also enter in the shoots with microbes, or plants cultivate microbes there and take them into the shoot cells as well, the root cells, and take them in. They actually go into the nuclei. At least some of them go into the nuclei, the nucleus. They go around the nucleus. Now, it doesn't appear that they always go into the center of the nucleus, but instead they go into the nuclear envelope, the little membrane that goes around the nucleus. And of course, all the DNA is in the nucleus. And we think that it's the microbes that are entering those nuclei that are actually causing the increased epigenetic expression in the plants. And that's where you get more amino acids and more phenolics and so forth. We think that those microbes, and we think it's probably, or it's possible, I should say possibly, ethylene and nitrate, because they both will cause genetic changes in plants. They'll cause plant nuclei to divide, the DNA, not the whole nucleus to divide, but the DNA in the nucleus to divide, and they cause this phenomenon called endopolyploidy. So they get more genes in their nuclei, and nuclei actually get bigger. And we think that phenomenon may be related to this increased nutritional expression that you see in plants. That's what we're really putting a lot of effort on. I have,
When you say they get more genes, are there like genes that are dormant that are coming alive, or are those actually creating new genes in the system?
Normally there's a lot of genes in a plant, any organism, right? They're not all expressed. They're not expressed at the same time.
Okay.
But with the microbes there, the expression of the genes is increased. And also because of this endopolyploidy phenomenon, the genes are multiplied, right? So instead of one copy of a set of chromosomes, you have multiple copies of those chromosomes in the same nucleus. So what that means is that instead of a little bit of arginine, you get a bunch of arginine, or methionine, you get a bunch of methionine, or lysine, or whatever it is. So with the microbes there, you're going to get a greater expression of genetics in the plant. You see the same thing if you look at organic crops versus more chemically produced crops. You see the increased genetic expression in organic crops. And we think it's this phenomenon triggered by microbes as they go around the nuclei and trigger gene replication and expression. It sounds very complicated, but it relates to the fact that the bacteria, the microbes, go right around the nuclei and influence expression, influence what those genes do, right? And what the plant does with its DNA. We're really fascinated by it because, first of all, we're trying to understand why these microbes, or what these microbes are doing around the nuclei and in the nuclei. So that's an important phenomenon. We like to see it and then figure out what the importance of that is. And so we can, once we see it, then we know we can fixate on it and just keep working on it, because you have a kind of a mental image of the phenomenon. Yeah. But it's really fascinating and very important.
Are you seeing any negatives to that too, I guess, on that side? I mean, if there's some
there's negative. There's negative. You want to hear the negative? The negative is they alter with the microbes there, they alter the mutation rate, the increased mutation rate in the plant. Okay, so if you're a plant breeder and you want to make a new cultivar, a new variety, one of the steps is to stabilize those traits, right? To stabilize the genome in the crop. So you get if it's corn, you get a corn plant that looks just like the other corn plant, right? Instead of instead of what happens when you have all the microbes there is you get corn plants that start to look all different. So you can't have an even monoculture. You get, some short plants, some bigger plants, some plants that have more nutrients, some plants that, in the case of corn cobs, if you're looking at the corn, the way the corn kernels are on the cob, you'll see mutations popping up where corn turns different colors and stuff like that, or it becomes lighter, whatever. It triggers transposons. So it triggers, the microbes are triggering genetic change in the plant. And so it's hard to get a variety like we like to have in modern cultivars. That's the downside. But it's also possible that we could create breeding programs where we use the microbes to increase the genetic variability in the crop. And when we get it the way we want, then we reduce some of the microbes to stabilize it. That's also possible. And then you have this kind of and then you can put the microbes back on the seeds, right? You put the microbes back on the seeds so that those crops then are going to have those microbes. So it could lead to a new kind of plant breeding using microbiology. It's interesting idea. Whether it'll work or not, that's irrelevant whether it'll work or not. It's a nice idea.
It's a heck of a question at the very least, to ask. Yeah, that's so, as related to that, do you feel like it's possible to determine, those things? Is that, determinable, what those microbes are doing in the nuclei and actually how they're applying themselves into the genetic expression?
Yeah, that's what we want to know. Exactly how they're doing it. Our hypothesis is that they're producing ethylene, at least ethylene, and maybe nitrate in some cases.
Okay.
We can see nitrate around there. We can, with our histochemical stains, we could see ethylene being produced. We can see some nitrate, so it's possible that those are the chemistry. That's the chemistry that the microbes are producing that triggers that genetic expression and the increased genetic expression in the plant. But of course, getting at that and proving that as a whole, having a hypothesis is one thing, proving it is another. So that's a bit trickier, but maybe we'll get there eventually.
This may seem like a dumb question, but when we're looking at, say, a GMO product versus a non-GMO, are you seeing any difference in, how those genes are expressed in these systems?
Well, in some cases I can't say, but in other cases we know that, for example, if you look at some of the modern corn types that have been through tissue culture and are perhaps they've been genetically modified. At least some of our modern corn cultivars. We don't know exactly what causes it, but what happens is they lose the capacity to associate with microbes, like, for example, mycorrhizae, VA mycorrhizae. They lose NOD gene. They're out of practice, right, for associating with microbiology, and so they lose the capacity. So those plants may not be redeemable, able to turn into more microbiologically competent plants, crops. But on the other hand, from the corn work, we know that we can take some of these conventional cultivars, conventionally grown, conventionally bred cultivars, and we can provide microbes to them, grow them in biodynamic circumstances, provide ferments that are full of microbiology and other sources of microbes, put them there, and we can turn those plants into plants that are capable of using microbiology again. So it may not be an irreversible process. It may be that we can take many of our modern cultivars and begin to turn them back into microbial rich crops. So it may not be a dire situation for us.
Well, and that's a good transition as we wrap up, this update. It's there is remediation that has to do, but it is imminently possible if we focus our systems. And the research you're doing are unlocking these concepts that I think we've seen hints of that were true, and really getting to what are the truths that are working in our systems. And it's amazing that we're so advanced in some areas of our science and understanding, and so unaware of how our food is made and what produces good food and nutrition. And it's just I can't say enough about how dependent we are upon researchers like yourself and your team. So please pass along our thanks to your team. Before we go, is there anything else you want to add to the conversation? And I do want to ask you about just to make sure, how's your garden doing this summer too?
Not so good this summer.
Not Shoot, so I didn't mean to put you on the
spot. I'll tell you why. I'll tell you why, though. Our backyard is really, we've got trees on one side, we've got the house on the other. So we have a narrow strip of, and then as the leaves come on the trees, we get less and less light. So we have to really in order to get a good garden, we have to get a very early start before we get all the leaves. And so that there's enough time, because we really only have about 7 or 8 hours of daylight. So we've got to have a long growing season to do that. So this year we got a late start, and we wanted to improve the soil, and so we did that. We improved the soil, but we didn't have enough of a season to really produce. So we got a few tomatoes, a bit of okra. Our watermelons were stolen by the squirrels. Mushmelons were damaged by probably squirrels again. We just didn't do it right this time. That's it.
No, it sounds like, well, the squirrels might disagree, it sounds like, but
they had a great time.
Yeah. That's wonderful. Well, thank you again for joining us. Again, Dr. White, it's always a pleasure. Denise, you want to take us home?
Yes, thank you. So again, Dr. White, really appreciate you and your work and everything you're doing. And the team behind you, we saw some of them working behind you as you were talking. So that's really amazing too. Is there anything else that you wanted to mention in the conversation that we didn't get a chance to ask you?
Well, I think we've covered most of it. The thing about research is it doesn't turn as fast as people expect. Problems that take time to work on, it takes a lot of time. And sometimes, it's years in developing, like the understanding of rhizophagy. We were years developing that. I mean, the cycle part, at least, and how the process works. Basically years. And so it doesn't move very fast. So if we had another meeting in another discussion in 6 months, I don't know if you'd hear much different from what I said today, right?
I might have a follow-up for you, if you don't mind, Denise, just asking real quick on that note. That's a really interesting note. Is there anything on the human health research side that you would love them to explore, or researchers to explore, that would help you in your work? Or are there other soil, health questions that you would, encourage other researchers to investigate, support your work?
Well, I think there has not been enough work done on the actual connection between soil health and human gut health, human health. I think that would be eye-opening, if we could have studies like that were, controlled and replicated as much as possible, to look at the soil health, plant health, human health connection. It's hypothesized and people believe it, and there's evidence that it's there, but we need more work. We need more work to convince everybody, I think. So yeah, I think those kind of studies are important.
Appreciate that. All right, Denise, now I'm ready.
I appreciate that. the thoughts and additional questions too. And Dr. White, you're certainly not the only one who is interested in that research and more direct connection. We hear that from people all the time. And so, yeah, if you're listening out there and you're working on a study that is more directly connecting soil health to human health, please get in touch. We would love to learn about your work and your research, and help to get that out to the world and connect you with others who are interested in it too. So really appreciate your time today, Dr. White and Ryan. Thank you all so much. And with that, we will just close out and say thank you.